So on the practical side of things, all this genetics knowledge, like purple and white flowered pea plants, comes into play where we consider that the process of inheritance is the same in all diploid organisms, including plants, animals, et cetera, including humans, right? And so probably if Mendelian genetics did not apply to humans, we would not be so interested in them. And so let's think about one of the directions I never really did follow up with in the previous video because I knew we were going to be doing this. And it has to deal with it's a neat exercise. It's on inference. And inference comes into play very significantly in our discussions of genetics because we can use it to determine the genetic status of individuals within a pedigree. And so by pedigree, I mean a family tree, and we're going to be doing exactly that. And so in the textbook, you're given the situation, I think this is in 8.5 or 8.6, I don't remember, 9.5, 9.6. It was one of those sections where you were given a black Labrador dog, right? And you knew that the black lab was either big B, or big B, little b, right? You can't tell because big B is dominant. If you're little b, you're chocolate, like my girl, right? I've got a chocolate lab, and her name is Nikumi. And the thing is, well, if you have a black lab, how could you possibly tell whether it's big B, or big B, little b? You can't tell. In fact, we do this thing where we say, well, if we don't know what it is, we know it's either big B or little b. We'd say big B dash, okay? So let's say you've got a big B dash black lab, and you want to know whether it's a homozygote or whether it's heterozygous, right? And the thing we do is called a test cross. So if you've got, let's say the black flower, the black color is dominant, okay? What you do is you cross it with a heterozygote, okay? So in other words, you would cross it with a known little b, right? So here's your big B, little b, or sorry, let me undo that. Undo your big B dash. You cross it with a little b, a known a known homozygote recessive, and you look at the offspring. You would probably go for several offspring. Labs are fortunately very prolific in the number of offspring that they produce. And if you had like many male pups, males are squares, the females are circles. If you have like 3 male pups and 2 female pups, if they're all black, okay, if all of them have a black color, then you might infer that, well, it seems still possible that it's big B. You don't know for sure because hypothetically this could be a big B, little b, and we just happen to pass on the big B gene to all 5 of the puppies, right? But if you were to do that, and if you were to come up with maybe 4 black puppies and 1 chocolate puppy, right? This chocolate puppy has to be little b, which means this parent has to be big B, little b, right? In other words, we can take out big B dash and replace it with a known big B, little b, okay? And by doing this, we're basically using information from the offspring, okay? The offspring information is allowing us to infer the genotype of the parental generation. Got that? Okay. It could work the other way. It could work the other way as well. Let me undo a whole bunch of things until I get rid of all my colors. Oops, redo, right? Let's say, let's see, draw, okay. Let's say we have black labs and brown labs and the puppies, okay? Now we want to know, is this female puppy a big B, or is it a big B, little b? Okay, well, there's no question about this. It must be big B little b because its mom was little b little b. Okay? It has to have received at least one little b gene, and therefore, because it's got the black color, we know for a fact that it's going to be a heterozygote. So in other words, we know for a fact that that one's big B little b, this one is big B little b, this one is big B little b. Right? The only ones that are really easy are the homozygous recessives, because homozygous recessives, the recessive genotype is always going to be homozygote. Right? When you have the recessive trait, no question, it's got to be homozygote, right? See that? Makes sense? Because if you have at least one copy of the dominant gene, you're going to have the dominant trait. Yep. Yep. So those are two things that we want to be able to do. Use parents to infer information about the genotypes of the offspring. Use offspring to infer genotype status of the parents. The other thing that we do in pedigree analysis is to use the overall pattern of inheritance to ascertain which trait is dominant versus recessive, because you might not always be told that, okay, well, black is dominant, brown is recessive. You might not have that information. You might just get information from a tree. You might get the information like what we have over here, and you have to determine from that whether the dark coloration here, the ones that are darkened in, that the darkened in trait is a dominant trait or a recessive trait. Okay, and that's also relatively easy. Okay, it's easy to do that because we know for a fact that if you have a recessive trait and it's mating with another recessive trait, in other words, if you have recessive crossed with recessive in the parents, you are never going to have offspring with a dominant trait, right? Because if you have big little b crossed with little b, all the offspring are going to have to be little b. There's no possibility of ever getting a dominant offspring, right? On the other hand, the reverse is not true. The inverse is not true. If you have a dominant trait versus a dominant trait, it is possible. For example, if you have a big B little b crossed with another big B little b, it is possible to get little b in the offspring, right? Can you see that in the Punnett square in your mind? So when you look at this pedigree right here, right? Can you tell me whether the darkened one is this dominant or is it recessive? Well, yeah, okay, it's not recessive, because if it were recessive, then two darkened in individuals would not be able to produce a non-darkened in offspring. So the fact that this offspring here is different from the parents, we know that this one is going to be the recessive, and maybe this one is the little h. And because it's little h, we know that both parents are big H, little h, right? So if you go to the textbook on pedigrees, maybe you got two parents, they both have widow's peak over here, widow's peak over here, and they have an offspring that does not have widow's peak. That's the hairline. Okay, if your hair drops down in the middle like this, between your eyes, that's a widow's peak. If your hair just this, and you don't have that little thing that goes down, this is a straight hairline. This is a dominant trait. This is a recessive trait, right? I like my diagram better than the pictures of the textbook. But in any case, if you have two widow's peak parents who produce a non-widow's peak baby, you know for a fact that widow's peak has to be dominant, right? And these two parents have to be heterozygotes. They can't be homozygous of big H, because if they were homozygous, then you would never be able to have a homozygote recessive offspring. See that? Okay, and so another thing you might be asked to do is to choose A, B, C, D. Are the genotypes that we have here, 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 and here? Do we know whether they're big H, or are they for certain big H, little h? Are you for certain that they're heterozygous? Or could they be either? Okay. What do you think? Well, I mean, you have to take into consideration that if you have big H, little h, if you have a heterozygote crossed with another heterozygote, okay, and you've got a heterozygous, or I mean, an offspring that has a dominant trait, it might be big H, right? Or it might be big H, little h. In other words, you can't infer anything about these two offspring right now. You would have to say that these are both big H, little h, or big H dash, okay? And if you have a mating between big H dash and another dominant individual, and they produce two dominant trait offsprings, further, you have no further information. These have to be big H dash, and this has to be big H dash. And this parent might be either homozygous or heterozygous. In other words, we don't have any information that would allow us to distinguish between the homozygous and heterozygous states for these other individuals with the dominant genotype, with the dominant trait, okay? Now, if, okay, let me see if I can do this. If this individual were white, right? In other words, if we actually had an individual in the offspring that was little h, okay? Then we could do the same thing. We could, if this was little h, then we could say for certain that this individual would have to be heterozygous. This individual over here, this female, had to be heterozygous, right? Okay, so it's really a matter of looking at the information that you have and using this information to make inferences about the status of either the generation up or the generation down. Okay. So way over on the right-hand side, I've got a practice for you. Okay. Tell me, is the colored circles, the orange trait, dominant, or is it recessive? Okay, well, I hope you said it was recessive, okay? Because if it were dominant and if the white trait were recessive, then we would never be able to get offspring that are orange from recessive homozygous parents, right? Okay. The only way for us to do this would be to have these guys be heterozygous. Okay, let's maybe make them big A, little a. And the offspring that are the other genotype, the other trait, would have to have the recessive trait, and they would be homozygous for the recessive gene. Right? These two offspring, we don't know what they are because they could be big A, or they could be big A, little a. So you would say these guys are big A dash, big A dash. But maybe we can get some information from this parent, right? About this parent by looking at its offspring, okay? But we look at that guy and we're saying, yeah, this guy has a dominant trait. It mates with a dominant partner and all of its offspring are dominant. Okay? In other words, no more information. These all have to be big A dash because we can't say for certain that they're homozygous or heterozygous, right? Because maybe it's maybe they're homozygous, maybe they're this individual could be either, right? And so again, if you don't have any information, you've got to be able to say, well, I don't have enough information. I've got to accept the idea that it might be either homozygous or heterozygous, right? Now, on the other side, we know for certain that all of these individuals that have the recessive trait have to be recessive homozygous, right? And this is the exercise that we always have BIO110 students do because it's a great way of reinforcing the skills that you guys are trying to develop as you're becoming more proficient in your critical thinking abilities. Okay, back to our conditions, right? So inference, important tool for pedigree analysis. We've been talking about using information from the parents to infer genotypes of the offspring, using information from the offspring to infer genotypes of the parents, using information from the patterns of inheritance to infer whether a trait is dominant or whether it's recessive, right? That was the last thing we did. Okay, next item for us, and this is going to be a little bit of a preview for the project that you guys are responsible for completing in the last portion of BIO110, which is on a genetically conferred condition in humans, right? The point here is that there are many human conditions that are inherited on the same basis of having a dominant trait or a recessive trait be the condition, right? In your textbook talks about some examples of dominant traits and recessive traits. You might choose one of those conditions for your project. I would actually prefer that you go and do some research on your own, find another condition that is not described in the textbook, and dig up information about that. Okay, but basically, if you use one from the textbook, you could say, well, an example of a dominant condition would be Huntington's disease, right? Huntington's disease is a form of neurodegeneration. It causes you to become really severely ill and die after maybe around the age of, the onset starts around the age of 30 or 40, and the decline is relatively quick. It's kind of like ALS, only its inheritance is much easier to understand, right? And so that would be an example of a dominant condition. An example of a recessive condition might be something like cystic fibrosis. Cystic fibrosis is as a characteristic that is carried by many individuals, and if you have a carrier mating with a carrier, if you've got a heterozygote mating with another heterozygote, a big A with another big A, there's a chance. Okay, you might not know that your carrier is of cystic fibrosis because you don't have any known family history, but maybe you don't have it come from a very large family. You don't have that much information about them, and it turns out that you're both carriers, and you could actually have an offspring that has cystic fibrosis. Okay, it's a pretty concerning condition. Okay, so this is the dynamics of these diseases. That's one of the things that I'll be asking you to cover in your project. Okay? And we'll be talking about the project in greater detail on the D lecture from this module. So 13D will be dedicated toward giving you information that will help you get started in your research or your genetic conditions project. Okay. So bottom line, right now, all I want to tell you is that the things that we've been talking about so far would apply to some of the diseases that we're talking about. Some, not all, some human conditions. Are as easy as that. Big A, or big A, or little a, and maybe easy sounds good to you. Keep in mind that easy comes with the other side of the coin, which is that easy tends to be less intriguing, right? I mean, people that are listening to your presentation might be more intrigued and more rapt if you were to talk about something in which they've got to think a little bit more, right? And so keep that in mind as you're deciding what kind of condition to choose. Okay, so the other things that are on our agenda, I think these are pretty important things. ABO blood typing. I do hope that you, as a future health professional or just a citizen with knowledge, working knowledge about your own state of health, are aware of your own blood type, right? You can be, you've got the ABO blood type. You could be type A, type B, type AB, or type O. Okay? You also have the RH factor, which means it could be negative or positive. And so you could be A negative or A positive, B negative or B positive, AB negative or AB positive, O negative or O positive. So there are really 8 possible blood types you could be having. It's a good idea for you to know what blood type you are. Okay? If not, next time you go to the doctor's office, have them test you. They'll be able to run a test and give you that information right away. Okay. It's good information to know if you're ever in a situation where you need a transfusion, or if you want to give blood. If you want to donate blood, they'll definitely check you before labeling the blood that you give them, because they don't want some bozo telling them that, oh, I'm a type O positive blood, when it turns out they're type B positive, right? That would set people up for like a huge catastrophic mortality later on if somebody with O positive blood were to receive that transfusion. Okay, so if you're donating blood, they'll certainly be testing your blood before they take it, and they'll also be able to tell you what blood type you are, so you'll know the next time you give blood or the next time you need blood. Okay, so little public health issue for you right there, right? But ABO blood typing, whether you're type A, B, AB, or O, is also caused by simple genetic condition. Only in this case, we've got 3 alleles. 2 of them are dominant. 1 of them is recessive. Okay, and so how do you label these? Well, we use the capital letter I for the dominant ones, and the lowercase I for the recessive, right? And so the 2 dominant alleles might be I superscript big A, or I superscript big B. Okay. And if you think of all the possible combinations that you could have for these 3 alleles, okay, you could be I big A, I big A. You'd be I big A, little I. These are both type A. Type A blood. You could be I B, I B, or you could be I B, little k, little I, and these are both type B. Okay. So what we're seeing here is that these 2 alleles, big I superscript A and big I superscript B, are both dominant relative to little I, because their trait is expressed whether they're in the homozygous state or the heterozygous state, right? So big I superscript A completely dominates little I, and therefore it's a dominant allele. Big I superscript B is a dominant allele because it completely overwhelms the effect of little I. Okay? If you are little I, you are type O. Okay? And if you are big I superscript A, big I superscript B, this is type AB. Okay? And so if you have the 2 dominant alleles, they're both expressed. And this is a situation that they identify as co-dominance. Okay, so ABO blood typing is often identified as a situation as co-dominance because we've got 2 alleles that are clearly dominant, 1 allele that is clearly recessive, and it's something that you should be aware of. And you could actually apply this in a pedigree. Right? So for example, if you are type A and you are a male and your spouse is type, wrong shape. Your spouse, your female spouse is type B, okay? Is it possible for you to have a son that is type O? Okay? Is that possible? Well, yeah, actually it is. I mean, if this is big I superscript A, little I, this is big I superscript B, little I, And only if these are the genotypes, you know that the type O offspring is going to be little I, little I. This is entirely possible. Okay? Okay? We're using information from the offspring to infer the genotypes of each parent, just like we did before. Okay? So the rules of using inference in the situations of pedigree analysis apply even here. Okay? Now I'm going to go to another board to talk about X-linked traits. And in the case of X-linked traits, remember that the X chromosome looks like this. It's a fairly large chromosome. The Y chromosome looks like this. It's a puny chromosome, right? So in other words, if you are XX, right? If this is you, in other words, you're a female. If you are XX, you actually have 2 copies of all the genes that are on the X chromosome. Okay? Okay? All of these genes are there in 2 copies. Whereas if you are XY, a lot of the genes that you have on the X chromosome are only there in 1 copy, right? So in the case of genes that are carried on the X chromosome, for males, you're effectively haploid for those genes. Whereas for females, so 1 copy of the gene on the X chromosome is what determines trait, phenotype. Okay. Phenotype is the other word. I forgot to introduce that, but determine what trait you have. And that's basically all I mean there, right? Whereas if you're a female, you've got 2 copies, in which case you have to determine whether you're like big A, or big A dash, or big A, little a, or little a, little a. You know, these rules apply. You know, these apply. Whereas if you're a male, you're either big A or you're little a, right? So that said, you look at a condition like hemophilia, okay, which would be on the X chromosome. We might represent the gene for hemophilia as an X chromosome with a little h on it, right? Okay, so this means you would have a recessive hemophilia gene on the X chromosome, right? And the X chromosome without the hemophilia gene is often depicted by a little plus sign. Plus sign means that you've got the normal wild type gene, right? If these are the 2 genes, this is the gene for X chromosome with the hemophilia gene. This is the X chromosome with normal gene. Okay. If you're a female, you could be X plus, in which case you're not hemophiliac. You could be X plus XH, in which case you're not hemophiliac. Or you could be XH, XH, in which case you're a female with hemophilia. Okay. If you're a male, you could be X plus Y, okay, in which you're hemophiliac, in which case you're not a hemophiliac. Or you could be XH, Y, in which case you're affected. This would be affected, right? And so I know that you've already read through this in the textbook, and so you're pretty well versed on how this is working. A couple of things about this. One, what this means is that males are going to be far more affected. Actually, let me rephrase that. Far more commonly affected than females. Why? Well, in order to be a hemophiliac as a male, you have to inherit the X chromosome with the hemophilia gene, okay? Which may be 1 out of out of 2,000, maybe 1 out of 2,000 X chromosomes in the entire world is going to cause you to become a hemophiliac if you're a male, right? So in other words, you might have a fairly low incidence. If you're a female, you have to get an X chromosome with hemophilia. That's 1 in 2,000 chance from one parent. You also have to get an X chromosome with hemophilia from the other parent, which is basically the same likelihood, 1 in 2,000. And so your chances of actually getting this would be 1 in 4 million. And so basically what we see is the incidence of hemophilia in females is practically non-existent, whereas the incidence of hemophilia in males is actually kind of notable, right? So let's see if I could come up with an example of a pedigree analysis in which we have hemophilia, right? So this might be an example where you've got a male mating with a female, and they have offspring. They might have several offspring, 5 babies, female, a male, female, female, and a male. And of these offspring, the first male has hemophilia, it dies, he dies, right? Next generation, this female mates with male, and they have offspring, another 5 offspring. And in their offspring, all of the boys have hemophilia and die, right? And the only survivors are the 2 daughters, right? And so this is a type of pattern that you might be paying attention to. And this is one of the things that you might be looking for. Is the condition expressed in males only? Right? If so, you might very well be talking about a gene on the X chromosome. We call these X-linked genes, right? Another example of this would be color blindness, right? And so color blindness is actually a pretty common thing in the human population. It's like 1 in 20 males is colorblind, okay? If 1 in 20 males is colorblind, that means that the likelihood of females would be far more rare. About 1 in 400 females would have red-green color blindness, right? We take a very simple perspective. So this is the incidence in males. This is the incidence in females. Got that? Okay, so here's a true story. Okay? Jeff, that's me. I'm not colorblind. Not colorblind. Spouse, not colorblind. Right? This is blue. See, I just proved to you that I'm not colorblind. Okay, now here's the thing. The spouse, my spouse has 2 parents. My father-in-law, not colorblind. My mother-in-law, colorblind. Ooh, right? So the question is, Jeff and spouse have 2 sons, right? This is actually true. Okay, what's the likelihood? I mean, what is the prospectus of my children having the same colorblind defect that my mother-in-law had? Okay, working this out. Pause if you don't want a spoiler. Pause the video and work it out because I'm about to tell you what the answer is. Okay. So if you're following along with me here, the colorblind characteristic is caused by an X chromosome with a colorblind gene, right? So it's, say my mother-in-law is XC, XC, which means that my spouse, not being colorblind, had to have gotten at least one X chromosome from her mom, and the other X chromosome from her father must have been the plus type, the wild type, that allowed him to see colors perfectly fine, right? And so the spouse is XCX plus. Now, me, I know Jeff, not being colorblind, you know that I'm X plus Y. And therefore, if we can work out, we can actually use a Punnett square to figure out what the likelihood is for the different combinations of offspring are going to be for both gender and for color blindness. Okay, so if we put Jeff gametes along the top, I could pass on my X plus, or I could pass on the Y chromosome. Okay. If I look at my spouse's gametes, she might pass on either the X plus, or she could pass on the X chromosome that she got from her mother. Okay. So this is a Punnett square. It's a little bit different from the Punnett squares we've been looking at in the past, but you can fill it in. X plus is one possibility. This would be a female that has no colorblind genes. We could have X plus XC, and that would be a daughter that is a heterozygous for the colorblind gene. Okay, we don't have to worry about either of these because I don't have any daughters, right? Not important. Not that daughters are not important. They certainly are. All I'm saying is that because you know that I don't have any daughters, we don't need to look at this side of the Punnett square. Okay. So I know that I pass along a Y chromosome. So if I have a son, they could have received the X plus chromosome that my spouse inherited from her father. That would be this chromosome right there. Or it could, the son could receive the XC chromosome that from his mom that came from, that she got from her mother, right? So this XC chromosome could be there. So what's the likelihood of either son being colorblind? Well, if you're looking at this, these two possibilities are equally likely. So there's a 50-50 chance. Okay? There's a 50% likelihood that either son is going to be colorblind. Okay? Good. Now, does this mean that I've got one colorblind son and one non-colorblind son? No, it doesn't. I could flip a coin and get two non-colorblind sons, right? Or I could flip a coin twice and end up with two colorblind sons, right? That is, both of those possibilities are entirely conceivable. They're entirely plausible. In fact, I could have three sons and all three could be colorblind, or all three could be non-colorblind. Okay? However, if I had a daughter, there would be really no chance that the daughter would be colorblind because they know that they're getting an X plus from me. Right? Okay. So we're going to be spending some time in our class working out some examples of pedigree analysis. This is something that some students gravitate to very happily, and some students are like, oh my gosh, I need some help doing this. But sooner or later, with a little bit of help, they become happy as well. Okay, so look forward to doing that in class with you.